Okay, we are moving on now to lipids, another of the four types of organic molecules that we're discussing. And lipids are kind of the bane of everybody's dietary existence. If you're trying to lose weight, oftentimes you have to cut lipids out of your diet, which is a tough thing to do because lipids, things like butters, and oils are what make foods taste good to us. So they're often hard to cut out of our diets because it means our foods don't taste as good. The number one thing that lipids do for us is they serve as the most common way in which we store energy for later use in the body. Everybody knows that we store energy or food in the body in the form of fat, right? In the form of lipids. If you look at the amount of energy we can store per mole of lipids, it's about 9 kilocalories. That's more than double what we can store in terms of carbohydrates. Okay, carbohydrates only store about 4.3 kilocalories per mole. Lipids store more than double that. So they're a much more efficient way to store our energy for later use. Be happy that you store energy in the form of fat in your body. If you stored it in the form of carbohydrates exclusively, you'd have to store twice as much carbohydrates as you do lipids, so you weigh even more than you do. Now, what exactly are lipids? Lipids primarily are very long chains of carbons bound to hydrogens, and usually there's a couple oxygens in there, but it's by far and away predominantly carbons and hydrogens. Lots and lots of carbons and hydrogens in very long chains. Okay. And because there are few to no oxygens present at all anywhere in these molecules, I mean, if there is an oxygen or two present, it's like 1 or 2 oxygens compared to dozens and dozens of carbons and hydrogens. The overall behavior of the molecule then is to be very hydrophobic. The presence of those 1 or 2 oxygens does very little to make it hydrophilic. It's primarily hydrophobic. We know this is true if you put a greasy, fatty pan into water in your sink. You will see all of the fat particles rise to the surface and form large blobs. They're hydrophobic. They don't dissolve in water. There are lots of different types of lipids. The ones we're going to concern ourselves with in this class include things like triglycerides, which contain fatty acids in them. Phospholipids, which we are going to see in a couple weeks, are contained in the plasma membrane that surrounds all cells. Waxes and steroids, which are the basic component used for the manufacture of many different types of hormones in our body. All right, let's start out with triglycerides briefly here and talk about them. Triglycerides are basically made up of a molecule of glycerol for starters. And a molecule of glycerol is 3 carbon atoms with their accompanying hydrogens. And each of these are joined via an oxygen to a fatty acid. And that fatty acid can be different fatty acids for each of these bonds. Now, fatty acids tend to be very long chains of carbons and hydrogens with a carboxyl group on one end. If you don't remember what a carboxyl group is, go back to the last 2 lectures ago where I talked about the different functional groups and refresh your memory. So here is one fatty acid with a carboxyl group on it, joined here to my glycerol backbone. Now, one thing about fatty acids, they usually are even numbered in terms of the number of carbons they have. So they'll have 4 or 8 or 12 or 24 carbons. It'll be some sort of an even number. It'll have a carboxyl group on the end. And when I take 3 fatty acids and join them to this glycerol backbone, I have just made a triglyceride. I'll give you some examples of triglycerides. The different types of oils you use to cook with, like canola oil, vegetable oil, corn oil, peanut oil, those are all examples of triglycerides. Fats also fall into this category. So things like butter or shortening are examples of triglycerides as well. Now, what makes fats different from oils is that fats tend to be highly, let's see here, unsaturated. No, I'm sorry. I have to think about that again. Fats are highly saturated, sorry, and oils are highly unsaturated. So now I need to tell you what that means when I say saturated and unsaturated. A fatty acid that is saturated has the maximum number of hydrogens attached. An unsaturated fatty acid has some double bonds or triple bonds between one or more of the carbons. And as a result of that, those carbons, because they've bonded with each other, they are not bonding with hydrogens like they normally would. So this particular triglyceride is not fully saturated with hydrogens. We could force some more hydrogens on there if we broke apart these double or triple bonds. So let's take a look at some examples here. This fatty acid right here has all single bonds between the carbons. Therefore, it has a maximum number of hydrogens we can stuff on it, and therefore it is a saturated fat. This fatty acid down here, you can see, contains a double bond right here, and because of the presence of that double bond, we're missing two hydrogens. As you can see right here, if I can erase this, there we go. We're missing two hydrogens that would be right here. And as a result of missing the two hydrogens that are right there, we have an unsaturated fatty acid. It does not have itself saturated with the maximum number of possible hydrogens. Okay, let's take a little closer look at saturated and unsaturated, because being saturated or not being saturated has huge impacts on how a triglyceride behaves. For example, in these saturated fatty acids, like the ones that we associate with being things like butter or shortening, these are solid at room temperature. They're fats, and they're solid at room temperature. The reason why is this. When I have a string of fatty acids that has the maximum number of hydrogens stuffed on it as possible, and it's completely saturated, those long chains of carbons with hydrogens are very straight, and so they can stack up on top of one another almost like Lincoln logs, like to use and make a log cabin, as you can see in this molecule here. And when they stack up one on top of each other in a very orderly manner like that, they crystallize very readily at low temperature, and so they appear solid to us like butter and shortening does. By comparison, remember again that unsaturated fats have at least one or more double or triple bonds between the carbons, like we can see here in this example. And the presence of those double and triple bonds, although they didn't draw it here correctly in this picture, they do show it correctly here, causes bends to occur. Okay, a bending in the carbon chain. Anytime you have a double or a triple bond, there's a bending in the carbon chain, and that bending means that now these don't line up nice on top of one another anymore. They kind of get all jumbled up together, and so we can't stack them neatly one on top of each other, and therefore they do not crystallize readily at low temperature. They stay in a liquid form, and we associate these with oils. Okay, fatty acids that are liquid at room temperature we call oils, and these are the unsaturated fats. These are the healthier ones for you, if you don't recall. Okay, oils are healthier for you because unsaturated fats are healthier for you than saturated fats are. Unsaturated fats, again, remember, contain fewer hydrogens because they've got some of those double bonds or triple bonds between carbon groups. Now I want to take just a very brief second and talk about trans fats. Trans fats are also known as partially hydrogenated fats. And these are fats that were originally oils. Okay, and remember the oils have those double bonds in them, and we have artificially broken apart those double bonds and added in hydrogens to try to hydrogenate them. And when we do that, the more hydrogenated, the more saturated we can make that trans fat, the more it behaves like a solid at room temperature. Food manufacturers love trans fats because they're cheaper than using butter or shortening in their foods. And by the way, if you don't know this already, saturated fats taste better than unsaturated fats. Butter tastes better than canola oil. We all know that, right? So what food manufacturers have done have said, hey, we found a way to take oils like corn oil, which is very cheap to buy, and manipulate it in the laboratory to stick more hydrogens on it so that it behaves more like a solid. And by behaving more like a solid, it's going to make our food bulkier, and it's also going to make our food taste better, but at a cheaper price than if we were to use butter. Sounds all great, except for that trans fats are associated with things like colon cancer, and raising your levels of LDLs, which is the bad kind of cholesterol in the blood. So eating lots of trans fats or eating a lot of partially hydrogenated fats in your diets is a bad thing for your health. And yet at the same time, food manufacturers for a long time have used them indiscriminately because they were cheaper and allowed them to make their food and still maintain a profit. All right, enough on triglycerides. Let's talk very briefly about phospholipids. Phospholipids are an interesting brand of fats because they are amphiphilic. Remember again, amphiphilic. Amphi means both, like an amphibian survives in both land and water. Amphiphilic molecules can have a region of them that associates readily with water and another region in them that associates readily with fats. Phospholipids are amphiphilic molecules. They contain a glycerol backbone in them, same backbone as we had in triglycerides, but two of those glycerols are bound to phosphate groups. If you don't remember what the phosphate group is, go back and remind yourself on those functional groups with what the phosphate group is. Here's an example. I'm sorry. Only one of them is bound to a phosphate group. I apologize there. Okay, one is bound to a phosphate group. The other two carbons are bonded to fatty acids. So we get something that looks like this. Here's my glycerol group. Here specifically is the phosphate group. And then we've got these two fatty acids. The fatty acids are highly hydrophobic. They do not like water, but they do like hanging out with other fats. The region here where the glycerol is and the phosphate is has a lot of oxygens present. And because of the fact that it has lots of oxygens present, this region tends to be very hydrophilic. So this molecule has two behaviors to it. It has a region that likes to bond with water, has a region that does not. These are used to create the cell membrane in cells, which we will see in a couple weeks. They're also a different version of this amphiphilic molecule that's kind of similar to phospholipids are used in detergents. Okay. It's a related molecule in that it's amphiphilic, but it's not the same as a phospholipid. Exactly. Next group I'm going to briefly talk about are waxes. Waxes are a subcategory of lipids. They consist of extremely long fatty acid chains that are bonded to long chains of alcohols. Alcohols are organic molecules that end in a hydroxyl group. Remember your functional groups again. So here is my long chain fatty acid, and it's bonded to a long chain alcohol, and together those create a wax, like beeswax or the wax that coats the outside of things like blueberries. Waxes are solid at room temperature. Again, because these chains are relatively long and straight, they pile one on top of each other very readily, therefore they crystallize at low temperature. Last I'm going to talk about, I'm only mentioning very briefly, are the steroids. Steroids are lipids that are composed of 4 carbon rings. Okay, so you've got, here's one ring of carbons, here's another ring of carbons, here's another ring of carbons, here's the fourth ring of carbons. We get that same pattern over and over again. Now remember again, organic chemists are lazy. They don't want to have to write down a carbon at every single juncture, but you know that if you see an intersection at each one of these intersections, it's assumed there's a carbon and its requisite hydrogens. So we've got these long or these large four-ring structures. This is a steroid. Cholesterol is the base molecule steroid. Okay, so this is right here, cholesterol. We can manipulate cholesterol by adding a few other functional groups onto it, a few other R groups, and we can make things like testosterone, estrogen, other types of sex hormones like androgen, estradiol, etc., etc., etc. That is it for lipids. We're going to move on next to talk about proteins.